Today, I'm going to talk about biocomputing. It's something that you may or may not have heard of, but it, to me, is the next frontier of intelligent computing. So, what I'm going to go through is what biocomputing is, what the different types are, why do we actually need to be doing this type of computing, the different types of neural intelligence and what there currently is in the world, some ethical considerations, and some other cool things that I've seen that I think you'll like. What actually is biocomputing? It's a really interdisciplinary field. It takes a lot of different people coming together to make this type of computing work. And it really fuses together ideas from biology and computing and biological principles to design entirely new types of systems that allow us to use and understand biological processes for intelligent computation. Why do we need to be creating these new types of biologically inspired or biological computation mechanisms? And it's really because of some of the things that we've already touched on. The fact that current AI is way too energy inefficient. We have to use large amounts of energy to train models on GPUs. And not only are they too energy inefficient, but they're too data inefficient. So if you look into the literature, we're seeing that we're going to be running out of data in the next 2 years. Companies have already said that they are running out of data to train on, but biological systems or biologically inspired systems are able to learn with a lot less data to do the same sorts of tasks. Additionally, we as living things, we learn from experience or something called online learning. And this is something that we still haven't been able to do computationally. So being able to learn online allows the system to be able to act more efficiently. And this is also what I mean when I say that you can have dynamic intelligence. So, for example, when you may have heard with Tesla cars, they don't always function in the way that they should. And that's because the world that we live in isn't like a training set. The world is dynamic. And biological systems, by the way that they function, are able to operate in dynamic environments. And using these types of systems will allow us to create artificial systems that can actually work in the real world. It's sort of two sides of the same coin here. We can either have biologically inspired computing. So it's still on a silicon chip, it's still computing code, but it's based off of biological principles. Either the neural network has a structure or its function is inspired by biology, like, for example, this image here of a spiking neural network, or you can actually use the biological substrate itself. So, for example, you can use a mushroom. You can see this picture here, and take the actual chemical and electrical signals from it to do computation. And it's this that I work on, not mushrooms itself, but biological computing, and this is what I'll kind of go into a little bit more. So there are different types of biological substrate that you could be using, and here are just a few. DNA. So we know that DNA can hold a vast amount of information, but also interacting DNA molecules with different types of ions can allow you to do computation. So people have allowed DNA to do things like math problems. Um, Alongside this, we can use neurons, so brain cells, to be able to use their electrical signals to do computation as well. And additionally, mycelium. So mushrooms have, think of them as the roots. They are able to interact with each other, interact with the environment. Mycelium also have electrical signals, just like neurons, to allow them to communicate. And by manipulating these electrical signals, you can also create intelligent systems that can do computation and also proteins. So in the same way with DNA, you can have them in a solution and interact with different types of ions to allow them to sense their environment and also adapt and change in ways that when you know what you put in, you can have them do computation. I'll talk more about neural intelligence because this is what I work on specifically. And within this, you can see there's sort of two paths. We can work on 2D cultures. So this means having a two-dimensional neural culture in a single layer. Um, and this allows you to look at cell-to-cell interactions. You can put these on what we call microelectrode arrays, which simply means that we have lots of little electrodes that can apply electrical stimulation and also read that electrical stimulation from the neurons. Top of this, we know that brains are three-dimensional, so we can actually take the 2D cells and actually grow them into 3D cultures. These are called organoids, and an organoid is just a small version of an organ. And these tend to actually mimic some of the structures that are seen in the brain. And what this allows us to do is develop more realistic models of neural activity, but also hopefully um, have more complex computational behavior. So I'm going to talk firstly about a company that is developing in 3D organoid space called Final Spark in Switzerland. I've been working with this for quite a while, and what you're seeing right now is through microelectrode arrays that have different numbers of neurons. So, for example, on the left-hand side, the organoid is in the middle, here. I'm not sure if you can see my mouse, but in the middle of this, we have that weirdly shaped thing. That's the organoid with lots of electrodes attached to it. So what this allows you to do is record and stimulate the neurons in a pattern that you decide to be able to, but allow it to do computation. And then on the other side, we have something similar, but instead of it just being a single a single organoid, we have four of them that you can stimulate and record from in succession. So you can do this in parallel. Really interesting that these guys have done. Show you on the next page.
You see here is not just a virtual butterfly in a video game. It's something far more extraordinary. What makes this butterfly special is that it's controlled by a lab-grown mini human brain, remotely connected over the internet. Sensory input and world information from this 3D virtual environment are transmitted to this brain organoid by specific electrical and chemical stimulations to mimic sensory input. This brain organoid, composed of 10,000 neurons grown in the laboratory from induced pluripotent stem cells, interprets the input and sends back its signals, controlling the butterfly's movements in real time. It's as if the brain itself is living within this simulated world. This is the first time this has been done continuously over the internet. This entire experience is powered by a WebGL simulation, accessible through any web browser, making a virtual world for the brain just a URL away.
So this is one such application, and like they mentioned, this is all available over the internet. So we're going to see a world very soon where people can use this in a similar way to Google Colab, but it's still very early in the research. We're also going to talk about the other side now. So not using 3D cultures, but also using 2D cultures. This has come from a company called Cortical Labs that I also work with. And they've kind of taken a different route where they have created a system that allows you to keep these neurons alive. So in the same way that we have clusters of graphics processing units or GPUs for doing AI computation, what they are moving towards is a world where we have racks of these microelectrode arrays with neurons inside of them to be able to do the same kinds of computation, but with drastically less energy and data demands. I'll show you a demo of one of their most amazing pieces of work where they were able to get these neurons, so living neurons, to play the video game Pong. This is the microelectrode array that you're seeing here. So each one of these squares you can think of as an electrode, and there's a few different things that you can play around with, like for example, the actual amplitude of the electrical signal that you're placing into the stimulation. And this on the right-hand side is the actual Pong game. So if I just leave the settings as they are, you can now see that these spikes are the electrical stimulation coming from this game. To explain it really, really simply, the position of the ball right here is then encoded into the stimulation that you're seeing in that top long rectangle. So when the ball moves, you get a pattern of stimulation, and when the to make the paddle move, you have this left section that will make it move left, and the right section that will make it move right. So depending on where the activity is greater, the paddle will move either to the left or to the right. And if you're familiar with something called reinforcement learning, either from psychology or computer science, what we do is we give it negative reinforcement when it misses. So that top panel receives a lot of stimulation that doesn't really correlate to anything in the game. And what the neurons will eventually do is learn, okay, do I make more activity in this left region or in this right region so that I don't get that negative stimulation. This is the general premise of how you would train any kind of living system to be able to do a particular task. So you can all try this. It's freely available, and it's just a simulation. So there's no neurons actually being stimulated with electricity here. A good example to see exactly how you can change different parameters to make the the paddle do something. There are some other really interesting things that have happened very, very recently. So, towards the end of last year, there was a research group in the US that also liked this idea of bio-inspired computing. And you'll see this term biohybrid come up quite a lot now. It's the idea of fusing together biology with computation to be able to do things. And they were able to use mycelium to drive the motion of a robot. So what you're going to see here now is an example of a mycelium controlled biohybrid robot. And this uses exactly very similar principles because mycelium also are able to produce electrical signals. And then once you understand how to either cause those electrical signals or what those electrical signals mean or how many different types of signals there are, you can then use those to drive the control of a system like a robot. And these biological systems are actually most suited for the control of things like robots than other tasks that would require AI. To show you this video now, it's sped up. Sped up. What you can see is the experimenters are using a UV light to stimulate the neurons, sorry, the mycelium in the Petri dish that's at the top of this robot. And they use a pneumatic controller. So it's just air being pumped in and out of the legs. But you can see now that mycelium is now moving the robot towards this light. The general idea is that the mycelium will want to move towards UV light, and that's basically what it did. While all of this looks really, really cool, there's still a lot of hurdles that we need to overcome to progress this field. So, for example, interfacing technologies. There's still a big problem in interfacing with neural systems or mycelial systems or anything biological for a long period of time. Applying electricity, then the electrodes will degrade if we're and also damage the cells. Organoids, like I mentioned, they're 3D, but right now all of the hardware is two-dimensional, so it kind of sits on top, and that means only one side of the organoid is actually coming in contact um, with computation with the multi-electrode array, meaning that you're not really gaining anything from it being 3D. And also they have to be in these very specialized devices. Yes, I showed an example of mycelium being inside a robot, but we can't really do the same with neurons because they need a lot of other things to keep them alive. So these are some hurdles on that side. But then also we know that if we're playing around with the biology, well, we're not just electricity. We have a lot of different molecules that interact with cells to be able to allow them to function. And we just don't understand how all of those molecules interact with electricity at this level yet to really be able to control and do really advanced computation. On top of all of this, there's a lot of ethical considerations that we need to really think about, especially when we're using neurons. Because, for example, organoids, they are biologically plausible. They are 3D structures that can form structures that are similar to our brains. So what does this mean about sentience? What does this mean about their ability to potentially in the future feel pain, potentially perceive the world? We are also unable to interpret their internal states. So they're quite literally a black box right now. We look at their activity and we say this activity is different when we do this versus do something else, but we don't know what else is happening inside. And as we develop more advanced organoids, that interpretability is also going to be harder to achieve. There's also a lot of talk about the potential of consciousness in these sorts of systems. Our brains are made from neurons, and the bigger that we make these systems, the more likely that they become in some way conscious, whatever that means. And with all of this, that therefore means that we need to have some way of having regulatory oversight and accountability for how these systems are actually going to be used. I hope that was interesting, and I'm open to questions.
Thank you so much, Robert. It's really, really fascinating. Yeah, I'm just going to open the Q&A. Question is it true that nanotechnology will replace humans and doing all the work by their algorithm? Or is this not even possible in today's conditions?
So nanotechnology that's actually something that does fit within this, but I don't think nanotechnology is going to be what replaces humans. No, I wouldn't say that.
Thank you. How does AI help scientists, doctors, and other people working in private sectors use the 2D and 3D cultures to interpret diagnosis and still visualize it with the knowledge of biocomputing?
So it's really interesting when you look at these from two different sides. So the 3D cultures, because they are more biologically relevant, they are able to form structures that are similar to the brain. You can have much more personalized medicines for people. So you can take someone's skin cell, for example, and reprogram that into a neuron, grow this into an organoid, and then test different medicines on that person's specific organoid to understand how it would impact them. With 2D networks, these are less biologically plausible, so you wouldn't really want to do anything like that with these. Really, they would be for computation. And there are people talking about the potential of these replacing things like convolutional neural networks that are used for image classification and potentially allowing them to help do diagnosis if you could encode your data. So let's say you had information about a patient and you wanted to encode your data as electrical stimulation. This could be one potential application to help do that kind of diagnosis in terms of classification. That's a lot more far into the future. Definitely not something that could be done reliably right now.
This next question kind of ties into the last question. How can biocomputing help people in the future?
So right now, I think the biggest benefit is the fact that it uses so much less energy and data. So we're seeing that AI companies are needing more and more energy to be able to train their models. And that has a knock-on effect on global warming, on the environment. So being able to use these systems that are using less energy is going to help everyone in the long run because, well, we'll be scaling our intelligent systems that everyone uses, but being able to do that in a way that is actually environmentally sustainable.
Thank you. What are the biggest challenges in developing biocomputers?
So, keeping them alive. So this is one thing. because, so for example, with organoids, they are 3D cultures of cells, and that means that they can have cores, so that the middle of them can basically die if you can't get nutrients into them. And that's one big problem. How do we keep these systems long enough to really understand how to do computation long term? There's also the ethical considerations that I mentioned. What can and can't you do with them? But that's more for the future once we figure out how to use them properly. Now it's how do we keep them alive? How do we interpret the signals from them?
If there are moral and ethical concerns, how can we possibly identify them?
I think with these sorts of systems, it's really about trying to understand, well, coming up with a kind of red line for what we should and shouldn't do as a start. So there is already something called the Baltimore Convention for Organoid Research, where a lot of researchers came together and essentially said, these, this is what we're going to use organoids for and what we're not going to use them for. So that's one thing. I think the research community is at the forefront of this technology and has the power to put these checks in place now. That's probably the best way of going about it. There's also the idea of consciousness and how do we even describe or measure this. And some people have started to look into mathematical models of this. So I think making sure that any type of research has this within the protocol to do this sort of measurement could be a good way of um, just making sure that we do things in the right way.
How can nanotechnology, bio, and neurotechnology relate to AI?
This sort of technology is sort of a alternative to AI. I wouldn't say it's an alternative to all types of AI. For example, it's not going to replace your ChatGPT. It's going to be doing anything that a reinforcement learning algorithm can do. So, for example, training a robot, for example, is definitely on the cards to be able to interact with this environment. So that's sort of how it links to AI, being able to replace these very energy intensive models. How does nanotechnology also link into this? So being able to deliver chemicals deep into the organoid or into the culture because right now, like I mentioned, we're only using electrical stimulation, but we also should be using things like dopamine and any other sort of type of neuropeptide that's present in the brain. Being able to do that in a targeted way, we we haven't got there yet, but we have methods of getting drugs into different tissues in the body using nanotechnology. So I feel like nanotechnology would be very beneficial to do that, being able to target very specific regions of a 2D network or very specific regions of an organoid with a specific chemical.
Thank you so much. Another person has asked, what about cancer? Will it work on cancer cells, or would it be in the path of future creations?
I'm going to assume that they mean would this sort of technology be useful if you were to use a cancer cell? They essentially proliferate, they grow uncontrollably. And that's an interesting question because I think you wouldn't want that. You wouldn't want a network that continuously grows. It's like I said at the beginning of the field. So whether you'd want to use a I mean, I have another thought. So you wouldn't want a network that continuously grows because that's not entirely beneficial. But then also, cancer cells are what you call immortal. So it would then speak to this idea of keeping them alive. That might be actually a way of not maybe using a cancer cell, but some kind of immortalized cell would be maybe better for long-term applications of biocomputing.
The person has asked, where could we learn more about how AI is being integrated into biological systems?
There's these websites. So if you go onto the Final Spark website, I'll go onto Cortical Labs website. Um, but really it's more into the academic field right now. So if you were to just Google biocomputing, Google things like organoid intelligence or biological intelligence or mushroom controlled robots, these sorts of things, the articles, the papers, the these sorts of things will come up, but there aren't any books on it, for example. There aren't any courses to teach it. It's really that's the sort of stage that we're at, where AI was in the 60s. That's where we're at now with biocomputing.
Amazing. And then because of time, I'm just going to finish off with one quick question. What is the future of this technology?
Really interested in trying to figure that out myself because, like I mentioned, it's not going to replace all bits of AI where AI is used. But I do see there being a lot of use cases where the environments are dynamic. So, for example, robots probably would be a very interesting and good use case. There aren't necessarily any people around, but you need to have an autonomous system that can figure itself out if it gets stuck. See that could be a very interesting use case, potentially in cases where you have a low amount of data that you need to learn from. So because they're able to learn off small amounts of data, this could be a good use case. So for example, maybe a production line, um, this could be something, or if you wanted to classify images, for example, because this has also been done, but you don't have a lot of images to use to use like a conventional neural network.
Amazing. Thank you so much, Robert. I'm going to close the Q&A now. I'm going to hand over to Favor.
Thank you for partnering with us and making this a reality. We are truly grateful. Foundation and Red Hat